you so much for joining us for today's session, Gene Therapy 101. We're so excited to have you all here for this really important session. My name is Kayla White, and I'm a Community Education Specialist at MDA. We're thrilled to have you join us for today's webinar. This learning series is part of our larger MDA community education programs, which focus on bringing the neuromuscular disease community together around education and resources. Be sure to visit the community education section on mda.org for updates on upcoming events. We are recording today's event and we'll be posting it to the website for on-demand viewing at a later date. And please know that all phone lines have been muted, but we will have a question and answer session at the end of the webinar. You can use the chat feature to send in your questions, and feel free to send those in as you think of them. They'll be addressed after the presentation. For over 70 years, MDA has led the way in accelerating research, advancing care, and advocating for the support of our families. MDA's mission is to empower people living with muscular dystrophy, ALS, and related neuromuscular diseases to live longer, more independent lives. Our mission comes to life through these 4 pillars. First, care. Through our national network of MDA care centers across the U.S., we support individuals and families from day one of their journey. Second, champion. Through our advocacy and education efforts, we empower families with information and resources. Third, catalyst. Through our investments in research, we are furthering advancements in treatment discoveries. And fourth, community. Through programs like Summer Camp, our Connections program, and our community events, we are helping individuals and families build supportive connections and relationships. If you haven't done so already, we invite you to join the MDA community by registering with us. MDA has invested over $125 million in the development of gene therapy for neuromuscular diseases. For decades, MDA has supported researchers who have worked to discover gene-causing mutations for neuromuscular disorders, developed and refined gene delivery tools and methods, and established protocols for safe and effective gene therapy. Just a few weeks ago, MDA announced the launch of our Gene Therapy Support Network. This program includes gene therapy resources and educational opportunities for families to inform their decision-making process, as well as opportunities to connect one-on-one with a gene therapy support specialist. It will also provide clinical networking to support ongoing clinical collaboration throughout the MDA Care Center Network. We encourage you to visit our Gene Therapy Support Network landing page to learn more and to download our resources. I would now like to introduce today's guests, Dr. Craig Zaidman and nurse practitioner Natalie Goedeker. Dr. Zaidman is board certified in clinical neurophysiology and neurology and is an associate professor of neurology and pediatrics. Pediatric neuromuscular nurse practitioner Natalie Goedeker completed her Doctor of Science degree in nursing practice at the University of Missouri St. Louis and provides direct patient care and is involved in gene therapy trials. Thank you so much for being here today, Natalie and Dr. Zaidman. I'll now pass the time over to you.
Thank you guys for having us. We're going to share our slideshow here.
Yeah, thanks for having us. We're excited to be here. Natalie, while you're getting this pulled up, why don't you tell everybody sort of who you are and what you do? And I know they introduced you, but why don't you tell them a little bit about yourself?
Okay, I am a pediatric nurse practitioner, so I have specifically done my training just in pediatrics, and I've worked in pediatric neurology for probably about 6-7 years, and then specific to neuromuscle for the last 4.5. I see children with neuromuscular disorders in our clinic. I help manage some of our patients who receive gene therapy, administer some of the gene therapies that require a kind of provider level person to administer them, like Spinraza for our patients with spinal muscular atrophy. And then primarily I work on our clinical trials as a sub-investigator, just meaning that I help you out in terms of managing the patients and the medical issues that come up to kids who are in clinical trials.
Aren't you the co-director? Isn't that right?
That might be true, yeah. That might be true.
Aren't you the co-director of the Pediatric Neuromuscular Clinical Trials Research Unit?
I think you're also the co-director.
I think. Anyway, we're both excited to be here. I'm a pediatric neuromuscle doctor. I've been doing this for over 15 years and under the tutelage of Ann Connolly when she was here with us, who taught me a tremendous amount, as well as Alan Pestrunk, who was our division head for a long time. So under their Phenomenal mentorship. I am excited to have Natalie join us, and we've been doing this together now for almost 5 years, and it's been a pretty good partnership. And hopefully you'll get a chance to hear our thoughts about all this. We're going to keep it informal and kind of burn through all of these this PowerPoint that we've got to hopefully cover a lot of ground, since we know there's a variety of different folks out there wanting to learn from us. So.
All right, so just to review kind of our general objectives for what we want everybody to get out of today, we want to provide some basics so that everybody can understand the kind of concepts behind gene therapy and what it is. Provide an overview of what developments are going on in gene therapy for neuromuscular diseases. Talk a little bit about the process of what it takes to participate in a clinical trial, how to access a clinical trial, how to access an approved gene therapy and what approved gene therapies are out there for neuromuscular diseases, and then discuss things that we think people should consider before they participate in a clinical trial or before they access an approved gene therapy product. Just kind of some thoughts that we have as we have managed people the last few years who've received these products, both approved and in clinical trials.
So let's start, Natalie. I'm sure you're ready. A situation you've been in many times. We get a first-time parents, and they've gotten a call from their pediatrician that their baby has tested positive for spinal muscular atrophy on newborn screening. So it's a child who thankfully was born in one of these blue states here, which purple blue states. That's fair. They are obviously distraught. They don't understand how this happened. No one in their family has had a disease like this before, and we get them in clinic within a day, sometimes two, and you see them, right? So that's the situation. We got a newborn baby with spinal muscular atrophy, and they did a little reading before they got there. They went to the MDA website, and they are all geared up about gene therapy. They want to know all about gene therapy.
Right. So when we meet a family like this, we try to kind of back things up a little bit. And so to review some basic terminology before we can get to treating with a gene therapy or talking about a genetic disease, some of the basics include what exactly DNA is. And so that's material that contains our genetic information, most of that located in the middle or the nucleus of the cell, organized into these things called chromosomes. And what a gene is something that contains the instructions that our body needs to make different proteins. So we have lots of different genes in the body, these little pieces that are on the chromosomes that give the body instructions to make different proteins that are needed. RNA is a copy of the genetic information kind of created from the DNA that helps turn those instructions that are needed to make the proteins. And then a gene mutation is a change to the DNA within our genes. You may also hear the term gene variant. And so what that can do, that actually happens to everybody, and it only sometimes causes disease. So we only find out about it when someone has a problem. And there's no blame behind it. It happens randomly. Sometimes it happens to as a result of things we're exposed to, and sometimes we inherit things from our parents. And these can change the instructions for how our proteins are made and function in our body. And there are a lot of different types of gene mutations or variants that can happen that can change the way a gene works, and as a result, change the way the protein is made or the protein functions that makes it not function in the way that it should. And so this just kind of shows some basics for how a gene works and what different things can go wrong. So there can be one letter that is different that makes the protein not function as it should. There can be a whole extra word or extra set of letters. There can be a whole set of letters that are gone. There can be something called a nonsense mutation where the protein basically just stops being made as it should. Anything to add to kind of these basics?
No, and one thing to think about is it's not just the type of mutation, but the effect that mutation has on the sentence. So you could imagine a change in one of the letters, but you could still understand the sentence. You could also imagine a change in the letter or an additional word that would totally change the meaning of the sentence or prevent the sentence from even continuing. And that is a big impact on whether the protein that's made is functional or not. And that's what causes disease.
So this just kind of depicts this a little bit more. So on the left, you can see the normal gene, kind of all the letters in the code lined up as they should, which the arrow kind of translates to the normal functioning protein. And on the right, there is one of these mutations that we just looked at. Something is different in this mutated or gene variant that causes an abnormal protein to be made that can't function as it should, or no protein to be made.
Yeah, and sometimes that abnormal protein actually causes damage. So you can have a gain of function where the protein is destructive, or you can have a loss of function where the protein either isn't made or doesn't work. So what is gene therapy? Gene therapy is a modification or use of genetic material in order to treat a disease. And it depends on the issue, sort of what the, quote, gene therapy approach is. You can attempt to replace a missing or mutated gene. You can add genes or turn off genes. You can attempt to edit genes, or you can attempt to modify the product of those genes. And when I'm teaching residents, it drives me up a wall when someone says, this is, so-and-so, and he got gene therapy, because that doesn't tell me what I need to know. And so we will use the word gene therapy, but what we're going to talk about is all the different things that means, because for patients, that having an understanding of what the drug or the clinical trial involves is really important, I think, and they're all very different. So what most people think about when they think about gene therapy is gene replacement therapy. So here, what you can see is a person on the far left of the screen who does not have a gene mutation, and then a person with a mutation where you can see they're missing gene 1, or gene 1 is non-functional, and so the protein just doesn't get produced. And if you're missing that protein, the absence of that functional protein causes disease. You need it. Your body needs that protein to function normally. And so if you could just replace that genetic information, then your body could make the protein, and that would help fix your disease. And so that's gene replacement therapy. So how is that done? How can you give someone a gene they don't have? Well, one way to do it is to use what's called a viral vector. A vector is a vehicle or an envelope to deliver a new gene into a cell. And for the most part, what we use are viruses. Viruses are really good at getting in our body and really good at invading our cells. I think of them like junk mail. They get in every house, they get in every mailbox, and not only that, but they trick you into opening them. And then when you open them, all that junk mail spills out, and you're like, oh man, I didn't want all that junk mail. So what scientists have done is they've used the envelope of these very effective viruses, the junk mail, but instead of having junk mail inside, what they're doing is they're putting in the genetic information that your body is missing. They're putting in what we call a cassette in order to deliver that information. And just like junk mail, you want the virus to go in the house where you want it delivered, except you don't want it to make you sick, right? You want you want your body to open it, and then you want to be excited about what's inside. You want it to work. What's important about this is that you can't have antibodies against these viral vectors. If your body has an immune response against the virus, the vector, the envelope, then you're likely to destroy it or have a reaction to it. And that is not good, And so we do have to be tested to see if your body has antibodies to the vector, and each potential gene therapy could use different vectors. Not all the vectors are the same, and as you know, they're making new ones, in fact, to target specific cells in your body and to try to avoid your body from making those immune responses.
And to be a little bit, when you say it's not good that someone has antibodies, we kind of mean is that first of all, it won't work in all likelihood because their body's already been exposed to this virus, so it will just kind of be cleared out. And also it may not be safe. They may have a kind of a bigger immune response than we would expect.
Yep, that's right. So here's an example of how a viral vector works. You can see there's the virus, like the junk mail envelope, and except inside it, instead of junk mail, is the functional gene. And the virus's job is to attach to cells and invade them. And it does that very effectively, ideally, and then it opens its products and it dumps the gene cassette in, which then has the instructions to make the protein that the person is missing, or whatever it is that the scientists have created in order to deliver this into the cell.
And the defective gene and everything else in the person's DNA just stays as is, correct? This
Yeah.
is not changing their DNA. It's just giving them this new thing that we hope will be functional.
That's right. So you can use these viral vectors to deliver a number of different packages, and we are going to focus on spinal muscular atrophy and a few other diseases relevant for the MDA community. But this idea of using what are called AAVs, or adeno-associated viruses, it can be used to deliver a variety of different payloads to the cell, and here are some examples of ways in which scientists are thinking about using them. And what I like about this graphic is it points out an important quality, which is that there are a lot of different organs involved, from the eyes to the brain to the spinal cord to the muscle, and different vectors have different affinity for different organ systems, just like not all junk mail goes into every mailbox. So that's part of the design of the product. Which viral vector are they going to use? And these new handmade viral vectors, these what's the word for it, Natalie? The
Engineered AAV, I think.
Yeah, these engineered viral vectors are sort of the new thing that we hope are coming to really target specific organ systems specifically. So
to go back a little bit to thinking about that baby that you mentioned with newborn screen positive spinal muscular atrophy. So just to briefly explain a little bit about what spinal muscular atrophy, or SMA, is. People with spinal muscular atrophy have a mutation on the SMN1 gene, or survival motor neuron 1 gene, which makes survival motor neuron protein. And that's a protein that the motor neurons, or the nerve cells, need, and the nerve cells communicate with our muscle cells and help our muscles function. And so without that protein, children get weak and their muscles get smaller, they get atrophied. Eventually, without that SMN protein, the motor neurons kind of break down and they eventually die off, and the muscle weakness and atrophy, it can't be reversed. And so this is a really exciting thing that happened within the last 5 years, is the first systemic gene therapy for a neuromuscular disease, or actually for any disease, that was approved by the FDA. It's called Zolgensma, and it is gene replacement therapy for SMA. So it's a one-time dose given IV. So the children have an IV placed, and we give it directly in their vein. And it's pretty miraculous. The new working SMN gene is placed inside this vector called AAV9. The viral vector, the kind of gene making up the virus that could cause any illness or anything worrisome is removed and replaced with this SMN gene. And the working gene is delivered to the motor neurons. This medicine, it's approved for all SMA types and children less than age 2. And we're participating or have participated in some clinical trials actually for some kids older than age 2, and including one that just ended. And then they're also looking at some trials not to give this medicine in the vein, but to give it one time via spinal tap in older children. We participated in one of those that ended a few years ago, and then there's a couple others that are ongoing.
All right, so the family's heard about that, and they're sitting there with you, and they are a little overwhelmed. And you've explained all that, and they say, we just give it right now. We want it yesterday. We're nervous, we're frightened, and we want to fix it. So just do it right now. We want it tomorrow. And how do you handle that? What do you tell them?
Well, we first kind of have to back up a little bit and talk about how there are other approved treatment options that also target spinal muscular atrophy, one of which is called Spinraza or nusinersen. And so we have to talk about all the options that are available to a patient so they can kind of decide the best thing. And so this particular medicine is also considered a genetic therapy. It's what's called an ASO, or an antisense oligonucleotide. And so this medicine, we didn't fully explain some of the genetics for SMA. I mentioned that there's the SMN1 gene is missing, but there's also another gene called the SMN2 gene that can make some of the protein that people with SMA need. And that gene essentially has one letter that's different in the coding from SMN1 that makes that gene only be able to make some of the protein that people need. And so the way Spinraza works is it targets that SMN2 gene and flips that letter back, essentially, so that people with SMA can use their existing genetics, the SMN2 gene, to make all of the functional protein that they should be making. And so that's the gist behind Spinraza. The difference between this medicine and the one-time gene therapy, other than how it works, is that it has to be given via spinal tap and it's given ongoing. It's not just a one-time dose. And then there's actually a third medicine approved for SMA that also is considered a gene therapy, and it's what's known as a small molecule medicine. It's given by mouth, so the children can take this medicine at home every day, just a liquid that they swallow. And this works the same way that Spinraza works. It targets that SMN2 and it helps that SMN2 gene make the protein that the children are missing. Yeah. And so what I would say to a parent for why we can't just give them Zolgensma today, which we would love to do, is that we first of all don't know if it's safe. We mentioned the antibodies that have to be tested for, and that takes some time. It's not just logistically, we can't order it to our lab and have them go on over and get a result in an hour like we can if we're checking a normal lab like a CBC or their blood counts or things like that. We have to send that out to a specialty lab and wait a few days for it to come back. We have to make sure to take those newborn screen results and actually confirm them. So we have to confirm their genetic testing with a specific SMA genetic test, just like any genetic disease that we would confirm. And we have to check some other things to make sure it's safe, that their liver is healthy and that their heart is healthy, and some other things that we consider before we just give gene therapy, along with the big insurance piece, which
the insurance piece too. So this is the SMA story is what I would consider a big success, and it also highlights an important point, which is that when you use the word gene therapy and you're talking about neuromuscular disease, it's not one size fits all. It's not just because there's a gene therapy for SMA doesn't mean that there's a gene therapy for a different disease. And you can see here, there's 3 different medications with their 3 different variations, all of which targeting the same disease, but using a totally different approach. And it's very specific to SMA, and in fact, very specific to the types of genetics SMA has. All of these are examples of having a protein missing from your body. So Zolgensma, which is a gene replacement, and then risdiplam and nusinersen, which are what we call splice site modifiers. So they take a gene that's very similar and they alter the splicing of that genetic code so that you can make a normal protein, a functional normal protein. That is similar but different for the newly approved tofersen, which is a medication for one genetic type of ALS or Lou Gehrig's disease. Patients with ALS from an SOD1 mutation, they have a gene mutation that causes them to form a protein which is damaging. So different from SMA, in SOD1 ALS, these patients make a protein that's harmful. And tofersen is also an antisense oligonucleotide, or an ASO, which binds to the mutated RNA, but unlike Spinraza, which creates a protein to be better than it was, in this case, the design of the ASO is to reduce the amount of protein being produced, and that's the way that tofersen works for ALS.
So I think all that said, when people say gene therapy now, I think they're under the assumption, a lot of people are, that we're talking about gene replacement therapy, replacing a missing gene, when actually there's lots of different approaches, as we've just gone over for just a couple diseases, and a little bit more to come.
Right. So let's go back to the baby with SMA. Natalie, you mentioned this, but just to review, is he a candidate for gene therapy?
So we don't know, just seeing him in clinic and doing an exam, we can't tell. We can't look at him and see, does this child have antibodies? And you would think, well, how would a newborn baby have been exposed or get antibodies to something? And in the case of SMA in a newborn, those antibodies are passed in the placenta from their mom. And so the assumption is that if a baby, when we test them, is positive, which happens probably 10, 15% of the time, maybe a little bit less, that those antibodies will decrease over time. So just because in the case of SMA in a newborn, a child is initially positive, the hope is that over time those antibodies will go away, which is not the case for other disorders. For older children, when we think a child is positive because they've been exposed to the virus, in those cases, we don't think the antibodies will just go away.
All right, and then there's all the practical considerations. We've got a family that's, living their own nightmare and emotionally fragile, and we're asking them to commit to a medication that once we give it, we can't take it away.
That's right, yeah.
And so we need to give everybody time to take a breath and learn. They need to learn about the risks of these medications, and we also have to make sure that the families are going to be compliant with the protocol, because this isn't just show up at the hospital, get the IV infusion, and go home. These children need to be on 2 to 3 months of steroid therapy. They have lab monitoring done on a weekly basis, and the relationship and the trust between us and them is pretty intense.
Yeah, I mean, the easiest part is what we do, which is not that easy, getting insurance approval for a multi-million-dollar medication. The hardest part is what the family has to do in the follow-up afterwards, and staying compliant with giving the steroids, so that we make sure this is as safe as it can be for the child.
Yeah, and in the case of SMA, we're in this phenomenal position where we actually have 3 medications to talk about, all of which work real well. And so there's some room for conversation and discussion with the families before we kind of, just jump forward. But we're going to jump forward. So why can't we use gene transfer therapy for all neuromuscular diseases? Like, not everybody on the call here has SMA. So you guys are all waiting. Like, what about my problem and my loved one's problem? Why can't we just do the same thing for them?
So as you kind of talked about a little bit with ALS, so not all genetic diseases have the same sort of cause. So not every genetic disease is a result of a lack of protein. Some genetic diseases have what's called a gain-of-function mutation that may not respond to just replacing a gene that's going to make protein. One other thing to consider is that different genes are different sizes. And so when we talk about the vectors that we're using to deliver the gene, we can't just put the gene in an IV and give it. We have to use this vector that delivers it where we want the gene to go. And the size of the gene can limit what we can do in terms of gene therapy. Some genes are really big. SMA is kind of a lucky one. The SMN gene is small and the full gene can fit inside the vector. You want to talk about the next couple? Safety.
Right, and then there's safety related issues. So when we dose these AAVs, they are dosed by weight. And so there's still risks associated with receiving these AAVs, even though they don't have the viral DNA in them. Your body still can have an immune response against these vectors, and that might be related to how much we give you. And so it might be advantageous to give this to a little person, but might be difficult to give to a bigger person or an older person. So all these things need to be considered and are being worked out in the clinical trial environment, and we are learning more every day. So
for a lot of diseases, the gene therapy it's not magic. It doesn't fix whatever damage has already been done. So that's something else to consider in terms of the uptake and how well it would work in someone and whether the risk is worth the benefit. Okay, so now to switch gears a little bit, we're going to talk a little bit about Duchenne muscular dystrophy. So thinking about this is another thing we see frequently. So we have a 5-year-old newly diagnosed patient with Duchenne muscular dystrophy, and they're coming to see us in clinic. And they're actually new to town. They just moved here from out of state, and mom and dad are still kind of getting settled. They're looking for work. They are living with family right now. And they want to talk about what treatment options are available for their son. We are still waiting to receive records, so we don't really know specifics other than we see this child in front of us. He looks like he has Duchenne muscular dystrophy. He's taking daily steroids, but they've heard that there is the possibility of maybe a new gene therapy for DMD, and they want to talk to you about what their options are.
Right, so we'll talk a little bit about Duchenne muscular dystrophy, and I'll try to highlight some ways in which this differs from SMA. The Duchenne muscular dystrophy comes from a genetic mutation in the dystrophin gene, which makes the dystrophin protein. It is the largest gene in the body. So unlike the genetic problem in SMA, here we're dealing with a very, very large gene. The protein being made by dystrophin resides in the membrane which surrounds the muscle fiber, and it serves to protect that membrane and sort of strengthen it. And so when a cell is contracted, it doesn't have dystrophin, the membrane tends to rip and the cell becomes damaged, and eventually the muscle will die and it will be replaced, but over time your body can't keep up and the muscle gets replaced with scar tissue and fat. So this is a pictorial example of the large DMD gene. In fact, these are what we call the exons, which are the parts of the gene that have the protein instruction in them. So actually, if you include the introns, it's even much bigger and gets even more complicated. And what you can see here is that the exons sort of are paired up. So you can see exon 1 fits against exon 2, and exon 3 fits against exon 2 and 4. And you can imagine if you were to remove or duplicate one of these, it might disrupt the way they fit. And if it disrupts the way they fit, then what we call that is we say it disrupts the reading frame. And that means that when you are trying to read this genetic sentence, your body gets stuck and it can't go from one exon to the next. And so the protein stops getting made right at that point, and that results in a non-functional protein, and that causes Duchenne muscular dystrophy. There's a version of Duchenne muscular dystrophy called Becker muscular dystrophy. It's also a mutation in the same gene, but in most patients with Becker, they are stronger because their mutation does not disrupt the reading frame. So for instance, they might be missing exon 4 or exon 47, but 46 can still communicate with 48, and so your body will make the protein all the way through. It'll just be missing that little part in the middle. So the reading frame would be preserved. So here is an example of what we were just talking about. Here, there's a deletion of exon 52, and that disrupts the reading frame. Exon 51 cannot fit against exon 53, and so the protein doesn't get made or is short and non-functional, and that results in Duchenne muscular dystrophy. And so here the idea is to, instead of trying to replace what's missing, is to actually remove another exon. And by removing exon 53, you can restore the reading frame, and 51 will talk to 54, and that will make a protein. Now, it will be missing a little part in the middle. It'll be missing whatever part of the protein exon 52 and 53 are supposed to make, but it'll at least go from start to finish, and the dystrophin protein, what it really needs is it needs the beginning and the end. And there are some parts in the middle that are variably important. And that's how some current medications for Duchenne are targeted. In fact, there are 4 approved medications that work this way: eteplirsen, golodirsen, Amondys, and Viltepso, and casimersen. I have a typo here, sorry about that. And they generate a really small amount of shortened dystrophin. Only about 1 to 5% of the muscle cells will respond to these medications, and we are hoping to do better. The community, the pharmaceutical industry, the scientists are hoping to improve that, but this is, at least it's a start. And hopefully some of the newer versions of these medications that are in trial will have improved protein expression. And then there are studies going on to use exon skipping or ASOs to target different mutations, including duplications and different deletions. Right now, these are approved for deletions that have to be in these particular areas. So you can see Exondys 51 is a 51 skip, and it only is approved and will only work for boys who have a mutation in which removing exon 51 will fix their reading frame, which is about 13% of boys with DMD. So you can see how knowing and understanding exactly the genetic mutation is a really critical part of caring for this family that showed up without all of the medical record information. I can't advise them about what their treatment options are until I know exactly what the genetic mutation is.
So the last question on that slide is why not just replace the missing DMD gene or the mutated DMD gene just like we do in SMA? But the challenge here is that, as Craig mentioned, the DMD gene is the largest known gene in the body. And as we talked a little bit about these vectors, we have to fit that gene inside the vector. And in this case, the DMD gene, the full-length DMD gene, rather, cannot just fit inside this viral vector. And you can see that in this picture here, the viral vector size is kind of on top there versus the DMD gene size on the bottom. And so the solution that people in the community have come up with is to select portions of the gene that we think are important to making a functional protein. We know it's not going to be the full length dystrophin protein, but we think it's going to function really well. And so they select those important portions of the gene to create a smaller version that hopefully can function in the muscles. And this concept is called microdystrophin. And you can see on the left here, they're pulling out these important pieces of this dystrophin gene to create a smaller version called micro or mini dystrophin, containing the minimum or so amount of the genetic code needed to make a functional dystrophin protein that's going to work in the muscle cell. And you can see this new microdystrophin that they have made now can fit inside this vector, which then are delivered just like Zolgensma, one-time IV dose. And they pick a vector that they hope is going to get into the muscle cells pretty well. And so the goal is to increase dystrophin production, in this case, micro or mini, a shortened dystrophin production, to then in turn hopefully improve the muscle strength and function and keep those muscles intact.
So this is an example of a designed or designer protein. You can see here on the left in those blue bubbles what a full-length dystrophin protein looks like. But in the middle there was a young man, actually was still walking at age 61, who had Becker muscular dystrophy and was found to have a really large genetic deletion. So was missing a whole lot of the middle of his genetic code. And based on his genetics, as well as studies of the way the protein works, it gave the scientists the idea that maybe they could make a really, really small version of dystrophin that would still function. And that is the critical question. So you can see there on the far right that the example of just how small this microdystrophin protein needs to be in order to fit into this viral vector. And so studies are being done to see if this microdystrophin can be delivered to the correct part of the cell and whether the protein is functional or not, whether it helps the boys stay or become stronger. And there are 3 clinical trials for gene transfer therapy in Duchenne muscular dystrophy, which you can see listed here. And everyone I can see in the chat is wanting updates on all of the clinical trials. And to be fair, it changes every week. Yeah. And so I do want to touch on some of the results from these, but I know the Solid Bioscience is, I think, going to be targeting an engineered viral vector. So they're pivoting a little bit toward trying to improve or optimize the way that their product is delivered. And Sarepta and Pfizer studies there have their phase 3 studies, which are ongoing. And it's worth, I think, talking a little bit about the Sarepta data, and we'll talk about the Pfizer data too, because the Sarepta data is currently in FDA review, and we have been getting a lot of emails from families all over the country, and maybe some of you on the web, that are curious to know about how this is happening. So the FDA is looking at approval of this product under their, what's it called? Their
accelerated approval. Yeah.
Thank you. Their accelerated approval pathway. And what that means is that Sarepta has asked the FDA to use a biomarker in order to determine whether the drug is likely to work.
And what's a biomarker?
And a biomarker is like a surrogate marker. So rather than say, do the boys get stronger, they're saying, well, do the boys make the protein? Because we know dystrophin, the more you make, the stronger you'll be. And actually, that's the key issue here. The FDA is like, well, yeah, you can show us. And in fact, Sarepta has done a good job, in my opinion, showing that the boys who get this drug make this little mini protein. I mean, they make it in 50% of their cells. The question is whether that little mini protein works. And
whether it translates to the the benefit that that 61-year-old had. It's
Yeah.
not the exact same. They engineered it based on his genetics, but they couldn't get all of the pieces that he had.
Yeah. That's right. And it's way different than the dystrophin protein that you make if you don't have Duchenne muscular dystrophy. So here is just one piece of data showing the challenge of this. There is only one placebo-controlled trial for this particular medication that has resulted. There is one that is ongoing and will be resulting soon, will be complete their first phase of it by the end of the year. And here you can see that in the younger boys, 4 and 5-year-olds who got this medication, in the purple line, you can see that over time they gained more function than children who got placebo over about a year. In the older children, the 6 and 7-year-olds, you can see that actually the yellow line and the purple lines look about the same trajectory. And you can also see that the boys that got the medication just because of random selection and who got put on the drug versus who got placebo were weaker by quite a lot, actually, at the entry to the study. And that mismatch in function makes it hard to interpret all of the data from this one study. And this is the issue before the FDA. And this is what they are wrestling with. Is this microdystrophin protein a good surrogate marker? Is it sufficient to say, hey, just because you make it, that the drug will work, to make you stronger?
Maybe we should just briefly say what this is looking at is something called the North Star test. And most of the clinical trials, no matter what the disease is, does something like the North Star test, which looks at motor function. And so they kind of target it and pick a test that's been tested to be valid as how we know boys without treatment will kind of change over time, which is what we see in the, well, no, this is
the yellow.
In the yellow.
Yeah, I see it in the yellow.
But yeah.
So we'll find out what the FDA is going to say like any day now. And we are almost as excited as those of you who are living with this disease and with children and loved ones who have it. Here is also some results from the Pfizer study. This is a 1-year follow-up cohort in boys who can walk. And what you're looking at here is you're looking at the change from baseline. In the green are the children who got the drug. And then here in gray, what they're showing you is an external control group. So not a group of children that were getting a placebo, but a group of children selected from other studies and natural history studies. And in Pfizer, Sarepta has similar data to this where they've done that. The FDA wants to see a placebo-controlled trial to best understand the results of the medication. And those trials are pending.
All right, so now to switch gears just a little bit and talk about limb-girdle muscular dystrophy. So this is a subgroup of muscular dystrophy. It is progressive. There are a lot of different subtypes, actually, I believe greater than 30, and each has a different genetic cause, which kind of leads to the challenge in this disease is that there's the need to target these different genetic causes, these different genes. Also, one challenge is that accurate genetic testing in limb-girdle muscular dystrophy is difficult. And often the genetic testing that we receive will come back with something called a variant of unknown significance, where the lab, it's just not been tested enough to show, does this variant that the person have really change the protein production? And we know that it does by caring for them and that this is most likely the problem. But before we would want to put them at risk and give them a gene therapy, we'd want to be sure. And then the other difficulty as we talk about limb-girdle muscular dystrophy and some other very rare disorders is that research is a challenge because there's not very many available patients. There actually, excitingly, are a lot of ongoing trials that have just gotten started, are in the early phases, or they're doing animal studies for these different subtypes that are listed here, 2E, 2D, 2B, 2I, 2A, and 2L. And to get into one specific example, this is looking at individuals with limb girdle muscular dystrophy, I believe 2A, who were treated with SRP 9003, which is a product from Sarepta. This is similar to the slide that Craig showed on DMD with the North Star scores. This is looking at a North Star test that is specific to people with limb girdle muscular dystrophy. And in the yellow, you can see the kind of natural history of how we'd expect that score would change over a 3-year period. So they have monitored these people for a pretty long time, versus the purple are those who were treated with gene therapy. And you can see that they had some improvement initially and then kind of plateaued or stayed stable over the course of the 3 years. There actually also is, not here in the US, but I believe this is in Germany, a CRISPR study, which Craig is going to talk a little bit more about the concept of CRISPR gene editing to correct one of the more common limb girdle muscular dystrophy 2A mutations. And this study was exciting because it works on what's called the satellite cells, which are responsible for making new muscle cells. And so this study, I think, was only done in about 3 patients or so, but the data looked pretty good in terms of the gene editing correction. It corrected in, I think, 2 of the 3 patients over half, up to 75, 90% of their satellite cells, which then corresponded to an increase in their missing protein.
So we have a lot of patients asking us about gene editing, and it is really complicated. And I have a very complicated slide I made here, which probably I shouldn't go too deep into it. But the short answer is it is gene editing, where there is a way in which you can cut the DNA and then allow it to repair itself, or even sometimes instruct it how to repair. The challenge is that it's hard to deliver that equipment. That CRISPR-Cas9 is what it's called. It's hard to deliver all of that equipment to the cells. So it can be done if you take the cells out of a person's body, and then you can modify those cells in the lab, and then you can give the cells back to them, is one way to do it. And another way to do it is to use that AAV vector and put into the AAV vector the mechanics needed to execute this gene editing, this CRISPR editing. And there are some safety concerns about this, like everything else, including what if it cuts the DNA in more places than you want, or it edits it in a way you didn't intend, and that can cause problems.
And same concept like we talked about with Zolgensma, once it's given, we can't take it back.
Yeah, can't take it back. And I think it's worth mentioning, because many of you probably heard about the young man with Duchenne muscular dystrophy who died about a week after getting the first trial of an AAV9 directed CRISPR therapy. And we feel this family's loss and the struggle in the community when something like this happens. And I thought it was important to mention him, and also to point out that he suffered some side effects, almost immediately, like a day after getting this. And probably, we don't know 100%, but probably he responded against the vector. He probably responded against the virus and not against the CRISPR activator, not against the CRISPR machinery, because he went downhill really quickly. And that's too fast for him to have had a consequence of the CRISPR itself. Probably he suffered from an immune reaction to the AAV vector.
All right, so now we're going to switch to another disease with some therapies. And we're going to talk about a family who comes to clinic. This has been an ongoing bit of a challenge for us, which is Pompe disease. And so this family comes to clinic. They have a 6-month-old son who was found to have Pompe disease on his newborn screen test. And they saw a doctor in genetics who told them that they think his genetic testing shows he has something called late onset Pompe disease. And parents say he's doing really well, and we agree. We examine him and we check his development, and everything seems to be going okay for him. They also mentioned that they have an older daughter, and she unfortunately is having some problems. She was just in gym class, had a little bit of trouble running the mile. So we decide to check her CK level, which is a muscle enzyme, and that came back high. And so then we sent some genetic testing showing that she also is positive for Pompe disease, the same mutation as her baby brother. So the family now, they're on edge about this, and they want to treat both their children as soon as possible. And they've heard a little bit about gene therapy, and they want to know if that's a possibility.
So it might be a possibility to use gene therapy in Pompe, but not yet. And what makes talking about Pompe, and the reason we included it in this talk, is because this is a real issue, a real challenge for how we counsel families. There is an FDA-approved therapy for Pompe, and in fact, there are two, and they are enzyme replacements, and they're given, through the vein every 2 weeks. But there are also, as you can see here, a lot of pretty new clinical trials trying to replace the mutated gene with a normal gene so that these patients can make their enzyme activity all by themselves, the enzyme that they're missing. And this issue with Pompe highlights, in this case with the baby who's clinically fine and the 16-year-old sister who's not, a lot of issues about Pompe. I did want to put in, since some of you out there may have Pompe, there are a number of currently enrolling gene replacement therapy trials. I have them listed here, but you can see that there are very few participants. 13 participants in one, 12 in another, and that's common for the early stage clinical trials for Pompe. There's even a gene therapy or gene replacement therapy trial for infants with infantile onset, but that's occurring in China. So this raises a lot of important points, which there is not a definitive answer. When you're talking about gene therapy, who is the best candidate? Should we give it to the baby before he has any signs of disease? Or should we give it to his 16-year-old sister who's early in the course but having symptoms? How long will it last? You can, at least right now, we can only give it once. And we don't know whether its effect is going to be permanent or whether it may wear off over time and then you can't get it again. There's a lot of risk involved in these clinical trials. People do die, and there is another alternate approved therapy available. So all of these things need to be considered by a family before they commit to one of these trials. And before we get into some of the details, I did want to mention myotonic dystrophy. So in myotonic dystrophy, it's even more complicated. And that's because there are many places along the pathway where one could intervene. And those are sort of listed here. You can, at the level of the gene for editing or transcribing it, or trying to inactivate the toxic transcript, or even trying to modulate what we call downstream effects, like more toward the level of the protein. And myotonic dystrophy is even more complicated because it affects multiple organs: brain, heart, muscles. And so when you're targeting therapy, you might have a success in improving hand grip, but you might not have any impact on brain function, depending on how the therapy is designed or targeted. And there are a lot of clinical trials that are targeting myotonic dystrophy, just like all these things we've been talking about. And so keeping up with this is challenging. And so we're going to talk about some ways in which you can keep up with it. But I wanted to mention that there's some early successes. This is a phase 1/2 trial in myotonic dystrophy with gene transfer therapy. And it's actually not gene transfer therapy, forgive me. It's a new way of targeting an ASO, and it has shown improvement in the grip myotonia that these patients experience in its early testing. So there's a lot to be excited and hopeful about in our field.
All right, so now to kind of wrap things up and talk about what people maybe everyone is applicable to everyone, I guess, accessing these therapies. So first, accessing some of the approved therapies that we mentioned, or some things that hopefully are coming soon. It is not a straightforward process. There's a lot of steps that have to be taken, including pre-administration. So you have to talk to whoever is treating you for your disease to determine if you're a candidate for whatever the therapy is that you're interested in. And there may be some fairly extensive testing, definitely since we're talking about gene therapies, genetic testing, lab testing to make sure that you're healthy enough to receive gene therapy, and also potentially including testing for the antibodies that we mentioned if you're talking about gene replacement therapy with a vector. Potentially cardiac testing to make sure that the heart is healthy at baseline. The insurance process alone can take weeks. These are not cheap medicines. It's not a medicine for your cholesterol that hundreds of thousands of other people are taking. So these are rare medicines for rare diseases, and not all insurers, when a new medicine comes out, they don't automatically have a policy in place for how they're going to cover it. And often there's very specific criteria that have to be met. What we look at when we're counseling patients and families is the patient and caregiver commitment. That's really what's most important to us, making sure that people understand what they're getting into. There's lots of lab work, and we'll give you a schedule of lab work and also say, but you got to expect that probably you're going to come in more often than that. If you're taking care of a newborn baby, you're probably going to have to bring your newborn baby in more than once a week, because it's not that easy to get labs in a newborn baby, for example. You have to take daily steroids for 2 to 3 months, and that is non-negotiable. And there's no guarantee that gene therapy, while we all have hope that it will help, there's not a guarantee of that in everyone. And in some cases, it could be harmful without a benefit. And then once again, as we've said a few times, we can't stop the medicine. Like, you can stop taking an oral medicine that you're taking at home. We can't stop it, and we can't take it back once it's been given if there is a harmful side effect. So some further kind of considerations for patients and caregivers. A detailed understanding of what the risks are. Most commonly in the approved gene replacement therapies, the most common side effect is nausea and vomiting. So usually pretty straightforward to manage. There can be some drop in the platelets, but usually that resolves after the first week or so. There can be a rise in the liver enzymes, because while we pick a vector that we think gets to the place we want to really well, we can't stop it from getting to some places we don't want it to go, and that includes the liver. And so that's one of the most common, sometimes more severe side effect that we see, is that the liver enzymes can rise. This can impact the heart. There can be inflammation to the heart called myocarditis. There can be more severe reactions, so systemic inflammatory reactions where the blood counts drop along with the platelets, and there can be some trouble with the kidney function. And when that's happened, that's been an emergency. There can be something called immune-mediated myositis, where the body is actually attacking the new protein that's being created. And unfortunately, a risk of gene therapy is also death. There's some considerations, which I'm going to get to, for siblings when there's an affected sibling with the same disease who maybe is not currently eligible for gene therapy, but could be in the future. And then also some considerations for patients' families are just that there's unknowns. Even after a medicine has been through all the stages of the clinical trial, because these are rare diseases, we see new side effects after approval that we haven't seen before. We saw that with Zolgensma. There's unknowns in terms of effectiveness in the specific patient and the durability, so how long the medicine may last. Again, the possibility of new or unknown side effects, and then that the current symptoms or damage that has been done may not be able to be reversed. And so this just kind of shows some headlines of what we worry about, these safety issues. And unfortunately, these have been some of the patients who have passed away following receiving gene therapy, including a patient with Duchenne muscular dystrophy in a clinical trial, along with the patient with Duchenne muscular dystrophy who received CRISPR that Craig talked about, a couple patients who received Zolgensma for spinal muscular atrophy, So moving on a little bit about the FDA process. So how does a medicine get approved? So the FDA mission, they are on our side. They want to make sure that the medicines that people have access to are safe and that they work. And there's actually a specific division within the FDA that looks at cell and gene therapies. And you can go on their website and kind of read through their stuff. I wouldn't, it's not that exciting, but for a new medicine to kind of get to a point where of like Zolgensma, where it's available for people with SMA who are under age 2, first they test new medicines in the lab and sometimes on animals, and then they move these medicines through different phases of trials in humans. And because these are rare diseases, sometimes the different phases are combined. One thing to think about in gene therapy is that there's something called dose escalation studies, where they have to test more than one dose to find out what is the safest and most effective dose. And so for a gene therapy where right now it's a one-time option, some people may get a low dose, and sometimes it may be too low of a dose to work, but that's part of how we have to test these things. And then there's something called a placebo-controlled trial where sometimes people get, for a portion of the trial, placebo. So they are getting an inactive medicine that it's not the gene therapy, and we don't know who gets what. And that also is part of telling whether the medicine works better than receiving nothing at all over a period of time. And then, as we talked about, there's something called accelerated approval, which is an expedited process. I don't know that we need to go into it. You kind of covered that, I think, enough already.
So maybe I'll talk about this. So many of you, and Natalie knows better than anyone, I think, are interested in clinical trials. And what I want to highlight is that being in a clinical trial is voluntary and it requires your consent. And that's the most important thing for us when we meet a family, because we know that you're fragile and we know that you are desperate. And we pause and make sure that you understand that participating in clinical trials is voluntary and carries risk and of unknown benefit. And we know many of you are willing to accept that, but it's a huge part of our job to explain it to you so that you have time to consider it. And not every person is a candidate. Just because you are interested, it doesn't mean that you can be in these trials. And we don't make those rules. The companies, the sponsors running the trial, they have very stringent criteria for who is a candidate and who is not. And that's part of our job, is to screen people for that and then help counsel them about which studies might be appropriate for them and which they're not likely to be a candidate for. And what Natalie was talking about was in terms of risk of being in a trial or risk of receiving a medication to a sibling or a loved one, is that when you do get one of these viral vectors, you do shed the viral vector. And we don't really know whether that can expose a household contact to the viral vector, and then maybe they'll develop antibodies against it, and then maybe they won't be a candidate in the future. We think the risk is low. Your body chews up these viral vectors, and so when you release it, you're not releasing the intact virus. You're releasing little parts of it, but it can be in your saliva or your urine or your stool. So it's worth talking to your doctor about that and about what you can do when you're caring for a baby, for instance. You know, we tell people to wear gloves when they're changing diapers for a month or two, and to double bag the diaper and throw it away so that the mom or the sibling doesn't get exposed to the viral particles. And I know what's coming for us in the all the questions, which is what's the latest and greatest for me. And what I want to tell you is how do I do that? Because I can't remember it all. Natalie remembers it all, but I can't. So I go to clinicaltrials.gov. And I have that written here, and I'm going to show you 2 slides of what it looks like. So you go to clinicaltrials.gov, and then you type, this is the first screen that comes up, and you type your disease right there where it says condition or disease. And I typed in Pompe, and then you hit search. And then you can see there's some filters on the left. And what I did was I clicked studies that are recruiting or not yet recruiting. And that way I don't have to look through studies that are closed. And you can see there that they come up and they tell you where their locations are, and you can click on them and read all about them. And I love it when families have found something and they contact me and they want to know whether they can participate. And that's an easy way to do it. So with that, we are into the question sessions by 10 minutes more than we should have been. And so I'm going to keep this real brief, because about 5 minutes before we came up here to do our web broadcast, I kind of did the smart list style where there's a little surprise. And I said, Natalie, you need to come up with the question for me, and I'll come up with a question for you. So she's been thinking the whole time about what she's going to ask me. Part of the reason I want to do this is because I want to set the bar real low, because I'm imagining we're going to get hit with some pretty specific questions we might not know the answer to. So Nat, here's my question for you. You field a lot of emails from people from all over the world about clinical trials. How do you manage that?
Well, I try to answer everyone, first of all. And I don't know exactly how to answer that question. I just do. I mean, I enjoy taking care of patients. I enjoy talking to people, and I know that everyone is looking for hope, as am I, in caring for patients. And so I don't know. It's not an option to not communicate with people. So we enjoy it. We have a slide that has our website, which has our contact on it. And I'm happy to talk to anybody anytime.
We did have a slide.
Yeah.
Oh yeah, that one.
Yeah.
But you can't give people medical advice over email or over the phone that aren't our patients,
That's
right?
true. I cannot give medical advice, but I can help connect them with resources like the MDA, like clinicaltrials.gov. I can connect them with a study sponsor if they have a specific study question. So there's a lot of ways to direct people without giving medical advice for their disease.
I'm so glad you came to work with me. You are a gem. All right, did you come up with one for me?
What do you think is most important takeaway for patients and families after what they've heard us talk about today? And we can also let people, there's a lot of questions.
There's a lot of questions, so I'll keep it brief.
Yeah.
I'll keep it brief. You need to have a very good relationship with your doctor. These kinds of questions and issues are too complicated for you to navigate them by yourself. They are too complicated for one care provider to navigate in isolation. Natalie and I are in constant communication with pharmaceuticals, with other colleagues around the world. We have lots of questions about what it is that we are doing. We talk with people all over, and we are trying to give you the best advice that we can. You should not feel like you should be responsible for navigating your own complicated healthcare. This is a gene therapy 101. If you don't have a doctor who understands, ask your doctor who could help you, and use your doctor to refer to a place like ours, we hope, that can be a trusted source for, frontline opportunity.
I think the most important takeaway is that there's a lot of hope out there for a lot of different disorders. And there were some new things that I learned making this presentation that I didn't know about for all the clinical trials that are ongoing, in spite of that's what we do every day. So.
Well, you answered your own question, didn't you?
While you were talking, I thought of my answer. All right. Should we stop sharing? Do you MDA, do you all have some slides that you were going to finish out with for the questions?
Yes, I will go ahead and start sharing my screen.
Sorry if we didn't leave quite enough time. We'll do our best.
That's okay.
15.
So a lot of the questions, you did kind of touch on some of the things about limb-girdle and stuff. So we're gonna skip past those just because you all did talk about those. So we're definitely not going to get to all of these, but we'll get through as many as we can. Do you know any good vector to make it to MDNA or where the scientific community is with that development?
Yeah, why don't you go ahead and start with a hard one, Kayla? Thanks a lot for warming us up here. Whew.
To get to
The mitochondrial DNA
Oh.
and mitochondrial DNA.
That's not a great question for us as clinicians and
not scientists. Yeah.
Yeah.
I don't know of one off the top of my head. I mean, I don't know the specifics of how the mitochondrial DNA would need to be replaced, how that would differ from the current therapies that are in place, because the mitochondrial DNA is different. So I can't answer that question.
That's a really good question.
It's a good question. I can't answer it. I'm sorry. I'd rather say that than guess and be wrong.
No problem. We've got plenty more for you all to answer.
Yeah, pick an easy one.
Let's see. The next one that came in was, can gene therapy be used in case of duplication of exons?
Potentially, yes. And there actually specifically are some ongoing or completed very small trials. The one that I'm thinking of is out of Nationwide, Ohio for Duchenne muscular dystrophy for patients who have a duplication of exon 2. And the idea behind it is editing out the extra exon 2 so that those children can hopefully actually make full-length dystrophin protein, so potentially even more promising than the gene replacement that we talked about. So that study, I think, was done in about 3 patients, but yes, that's potentially a possibility.
And a person with a duplication with DMD, for instance, that it has an out-of-frame mutation, that you could get gene transfer therapy.
True.
Right?
Yeah.
We're talking about specific therapies targeted to the duplication. But
Right.
Gene transfer therapy with microdys, you could be a candidate, depending on where your mutation is, you could be a candidate for that potential therapy. So there is no simple answer in the world of genetic therapies. It's all targeted to the specific mutation.
Okay. So you did talk about this a little bit, but just because it's so much information, I think it's important to hit on again. What kind of testing would you have to do before you undergo the gene therapy?
Let me take it because you already talked about it. The first thing is the family needs to have a conversation with the doctor regarding the risks of the therapy and the commitment. Once the family understands the risks of the therapy and the commitment in terms of other medications they'll have to take and the laboratory testing process, then the testing that has to be done, you have to confirm the diagnosis. So if there's any debate regarding the diagnosis, that is a stopping point. You need to, at least for now, we need to know the genetic, it has to be a genetic diagnosis for the ones I'm thinking of. And you have to be tested to see if you have antibodies against the viral vector. Other than that, we and others screen to see if you have other underlying diseases. If you suffer from liver disease, if you suffer from heart disease, that may make you, that puts you in a different risk category. If you've been exposed to hepatitis, that puts you in a different risk category because these medications can inflame your liver. So we screen for underlying diseases like hepatitis. And we screen your kidney function, we check your heart function, we check your liver function, we check your blood count. So that's the basics of it.
Okay, thank you. Somebody has asked, why would you need steroids with treatment, with the gene therapy treatment?
So steroids can help, they suppress the immune system. And so even though we mentioned that the vectors that we use remove the kind of genetic material that can cause an illness, the vector can still cause a immune response in the body. And so we try to prevent that immune response by treating with steroids for a couple months. As we are monitoring pretty frequent labs in people who get gene replacement therapy with a vector over the first 3 months or so after they get the treatment. Sometimes we have to do higher steroid doses to treat some of the side effects. One of the things I mentioned was that the vector sometimes goes places we don't want it to, like the liver. So there can be inflammation of the liver or an immune response. And we have to treat with higher steroid doses sometimes to get that under control, and sometimes do steroid in the vein and IV steroid for a few days as well.
Okay, so the next question is, do gene therapy changes carry to your offspring?
That's a great question. We don't think so. The gene therapy, when the virus releases its content in the cell, if the cell replicates, actually, it will not replicate that new DNA product. It doesn't go in your DNA. It lives next to your DNA. So actually, one of the challenges of gene therapy is if you deliver it to a cell that is constantly replicating, like let's say like growing hair, for instance, it'll only work until that cell replicates. And once that cell replicates, divides and replicates, the genetic payload won't be in the new cell. So we don't think so. It shouldn't target your sperm or eggs. For CRISPR-Cas9, which does edit your actual genetic code, theoretically, I guess it could edit the genetic code in a sperm, which are being produced. Yeah. Eggs are all produced already. Women, all the eggs you have when you're born. So unless those eggs are specifically targeted, I wouldn't think so. But if they were targeted on purpose or accidentally, it could. But the short answer is no, not right now, and we don't think so. Right?
Yeah, I mean, although of course we don't know for sure, it hasn't been studied, but yeah, we don't think so. The mechanism for how they work. Yeah.
Okay. Is the part that's affected by DMD considered the extracellular matrix? And they said, I think that's the correct term. So
that's a great question. And the answer is the dystrophin gene bridges the membrane of the muscle fiber to connect the inside part of the muscle fiber to the extracellular matrix. So it works with another complex called the dystrophin complex, the dystrophin protein complex, in order to bridge that gap. So the dystrophin protein is really important, and it connects to it, but is not part of the extracellular matrix.
Okay.
The dystrophin complex, that's what I'm trying to say.
Is there an approved gene therapy for the FLNC gene? And we're getting a lot of really specific questions for very specific diseases. So I don't know if you all want to kind of cover that and maybe go over again how people can kind of go through and look up what may be available for
which gene. Which gene specifically? I'm Googling it.
This question specifically was FLNC.
I don't know of anything specific for that gene, but we would direct, I mean, and what Craig mentioned that we do when we see something very rare, I mean, we don't have all of these things memorized and we can't keep up with all of the things that are ongoing. So when we see something very rare, we go to clinicaltrials.gov before we see a patient, and that's what we would suggest that all of you do. Clinicaltrials.gov, and you can type in what your disorder is and search for if there's anything available. It's required by law that the trials be listed on that website. So
yep, and before they're recruiting, in fact, they're listed on the website. So that's what I would do. I don't know of one off the top of my head, but we don't know everything. Natalie knows a lot.
Let's see. You did talk about this again earlier as well, but we got a few questions. So I just want to go ahead and have you all give you the chance to go over it again. A lot of people are asking about treatments for the older kids. Could these therapies apply to boys of 25 years of age with DMD and things like that? So I didn't know if you might want to touch on that again, since we did get a lot of questions about that.
Let's talk some specifics first. Why don't you tell them what people are trying, and in terms of what the studies are, who they're in.
This is asking about DMD.
We assume DMD.
Okay.
Yes, that question was specifically for DMD.
Well, my guess is that the question is potentially targeting a recent change in one of the Sarepta clinical trials, although I don't know for sure. But no, there is not a specific reason why gene transfer therapy could not be given to an older boy with DMD. The worry is that it could be less effective than giving it to a younger boy who has more healthy muscle cells. But no, I mean, our upcoming plan, we participate in several clinical trials, including one that is starting soon for non-ambulatory boys with Duchenne muscular dystrophy. And yeah, there's...
And Sarepta, the SRP-9001 has been given in a boy, I think, 20 or just
true.
over 20,
Yeah,
right?
1920.
Yeah,
Yes.
and the Pfizer study went up to...
To adolescents,
I can't remember exactly, 14 or 17.
But included non-ambulatory older boys.
Yeah, so it's coming, and at least our personal experience has been that the pharmaceutical companies are aware and interested in treating older boys with Duchenne muscular dystrophy. The challenge is, what do you measure and what would be considered a success? And that's an important consideration when you're designing a trial.
Thank you. So we are coming up on the end of our time, but I think we can get through a couple more. So somebody has asked, I often hear genetic drug using AAV as a vector to deliver into the body, but I also heard that it won't work for adults because most adults have already been infected with AAV. Would you help me understand this? If a genetic drug uses AAV as a vector, can it apply to adult patients?
This one's for you, Natalie, first author on this paper.
Well, there actually is not just one AAV. There are different subtypes, and the different subtypes have different exposure amounts, I suppose, in the community, or different percentages of people who've been exposed and are positive in the community. And so it depends on the specific subtype. And no, being an adult does not automatically mean that you have been exposed to whatever specific AAV is being tested.
Yeah, and in fact, the selection of the AAV, it's designed to be safe. It's designed to go to the target cell, and it's designed to have a low immunogenicity in the population. So the scientists are trying to find a vector that actually most people do not have antibodies against. And these engineered vectors, which are vectors that are really being built, are also designed to not have antigenic regions or regions that trigger an immune response on them. So being an adult does not exclude you from potentially being able to receive one of these. You need to be tested to see if you have antibodies. And just like a brand new baby needs to be tested. That's the only way to know.
All right. I think we've got time for this last one. Somebody's curious how long gene therapy has been around, and if there's any idea how long it's effective, or if it's hoped that this is permanent, or if that's yet to be determined.
The first systemic US FDA approved gene transfer therapy was in 2019 with Zolgensma, and previous to that there was one that was intraocular, if I remember correctly. People have been trying this approach for longer than that on a research basis, and I don't know exactly how long, but a very long time, 2 decades or more. So in terms of how long it lasts, the sky's the limit. It depends. The cell has to survive. If the cell that receives the transduced gene, the cell that is transduced dies or replicates, it will not pass on the drug, essentially. But for like a nerve cell, which never replicates, it's an excellent target tissue. Muscle does not turn over rapidly if it's healthy. So it's also an excellent target tissue for this kind of therapy. And hopefully will last a long time.
That's the hope. All right. Thank you all. We don't have time for any more questions. I know we didn't get to everything, but we want to respect everybody's time. So again, I would like to thank you, Dr. Zaidman and Natalie. Thank you so much for being here. This was so informative, and we so appreciate your time, your expertise, and everything you do for the neuromuscular community. We are so happy and grateful that you were here with us today.
Thank you for having us.
Thanks. I hope
it's been great. Yeah, I wish we had more time for questions.
I hope you guys liked it. We tried real hard to put this together for this broad audience. I hope it was a success. We really wanted it to be good. I hope it was.
It was very good. Thank you so much. We would also like to thank our interpreters today from Azuli Language Solutions. We appreciate their time and expertise as well. As gene therapy continues to change and advance, we encourage you to keep checking our Gene Therapy Support Network landing page for new information. Through our website, you can set up a video call with our gene therapy support specialist, download educational materials, and more. If you're new to MDA and are diagnosed with a neuromuscular disease under MDA's umbrella, or are a loved one of someone who is diagnosed, we encourage you to stay engaged with MDA. You can do this by visiting mda.org/join and completing a short form. Joining is free, and we'll keep you up to date on the latest research, educational programs, and supports. We would love to hear your comments about this webinar. Once the webinar is over, a screen will pop up with a survey. We would appreciate you completing this. This helps us ensure we are providing programming that meets your needs. If anyone has questions after this webinar, please feel free to email them to resourcecenter@mdausa.org or call us at 1-833-ASK-MDA1, and we will follow up with you. This concludes today's webinar. Thank you so much for attending, and we hope to see you again very soon. Thank you.